Bismillah Rahman Rahim. Welcome, dear student. This is Biochemistry 2, Chem 342 code. Today we are having lecture 21, and today's topic is cofactors of enzymes. This is your teacher, Dr. Naeem Khan, Associate Professor, Department of Chemistry, CUST. In today's lecture, we will talk about the various cofactors of enzymes. Enzymes require different types of cofactors for their activity. Without the presence of these cofactors, an enzyme is unable to carry out its catalytic activities. In most cases, enzyme needs these cofactors. So we will highlight about the details of cofactors. Then we will discuss the important types of cofactors and their roles with the enzyme activity will be highlighted, and the different examples of each type of cofactors will be discussed, and it will be pointed out the role of each cofactor they are playing in the enzyme catalyzed reactions, which is helping the enzyme to catalyze the reaction. So let's have a start. Chemically, we know that enzymes are of two types. Enzymes may be simple protein enzymes. And simple protein, we mean that those enzymes which are made up of only amino acids, because about protein we know they are made up of amino acids. Amino acids are the building blocks of protein. So if an enzyme is made up of only amino acids are linked by peptide bond, no other things. Such enzymes are called simple protein enzyme. And only few enzymes are such which are not require any other things and which are made up of only amino acids. For example, urease, amylase. These are only few enzymes. So enzymes are of two types. Simple protein enzymes are only few enzymes which are made up of only amino acids. The examples include urease, amylase, etc. But majority of the enzymes are complex protein enzymes. Complex we mean they are the enzymes which are made up of proteins plus non-proteinous part. Mean in these cases, the enzymes are made up of amino acids. They are chains of amino acid, but there is a part which is non-proteinous part. Means other things other than the amino acids will also be there. Most of the enzymes are like this class, complex protein enzymes. Examples are glucokinase, carbonic anhydrase, and so many others. The metabolic enzyme we know are complex protein enzymes. So first thing is, chemically talking, enzymes are of two types: simple protein enzyme, which are only few, and they are enzymes which are made up of only amino acids. But majority of the enzymes are complex protein enzymes which are made up of protein and some non-proteinous part, and these are the all major enzymes we know so far. In case of complex protein enzyme, the protein part of the enzyme is termed as apoenzyme. Apoenzyme is a word used for the proteinous part of the complex protein enzyme. The non-proteinous part of the enzyme is known as cofactor. What is cofactor? Cofactor is the non-proteinous part of the complex protein enzyme. Complex protein enzymes are the enzyme made up of proteinous part known as apoenzyme and non-proteinous part which is called as cofactor. And apoenzyme gets associated with the cofactor then we get a term whole enzyme, and whole enzyme is actually the ectoenzyme. In the absence of this cofactor, enzyme is inactive. So what is cofactor? Cofactor is the non-proteinous part that is required for the enzyme activity. Majority of the enzyme requires a cofactor for its activity. Generally, the cofactor represent a smaller part of the enzyme, usually up to 1% of the apoenzyme. Majority bulk of the enzyme is apoenzyme, and a small part is usually the cofactor, means the non-proteinous part. Cofactors are linked to the apoenzyme or remain associated for activity. It is not necessary that the cofactor will be always tightly bound to the proteinous part or apoenzyme. It may be just associated for the enzyme activity, or it may be tightly bound as well. Without the presence of a specific cofactor, an enzyme is totally inactive. Remember, with the exception of few simple protein enzymes. Majority of the complex protein enzymes are such that they need a cofactor, and if you remove the cofactor, the enzyme will be inactive. Cofactors, depending upon, cofactors may be organic molecules, they may be inorganic metals or inorganic other groups, or they may be metal ions. So cofactors may be organic or may be inorganic or metal ions, depending upon the type of enzyme. Cofactor helps the enzyme activity either directly or indirectly. We will highlight in the later slides that how a cofactor help an enzyme to carry out catalyze the reaction. So sometimes it is involves directly by accepting hydrogen or by removing hydrogen, or sometimes by accepting electron, or sometimes they are involved indirectly, how they are interacting the substrate to properly orient them to get attached to the active site. Active site, that site where actually reaction is carrying out, where is a catalyzing reaction is taking place, is still with the apoenzyme part. Active site is not with the non-proteinous part, we must remember. Types of cofactors. Cofactors have different types. A cofactors may be metal ions. We know that metalloenzymes are a very important class of enzymes. All enzymes which are requiring metal ions as cofactor are called metalloenzymes. And in this case, the cofactor for some enzyme may be metal ions. They are inorganic cofactors. Similarly, cofactors of enzyme may be organic molecules. Generally, they are termed as coenzymes. But among the organic molecules, they may be such organic molecule, they are just co-substrated, means they are transiently associated with the enzyme for catalytic activity. They are termed as co-substrate, while the coenzyme, which are tightly permanently bound to the enzyme for the catalytic activity, and they are termed as prosthetic groups. So cofactors may be metal ions, inorganic cofactors, or they may be organic molecules known as coenzyme. Among the coenzyme may be co-substrates when they are transiently associated with the enzyme for a catalytic activity, or they may be prosthetic group if they are permanently bound to the enzyme. Some types of the cofactors. Let's first type is the metal ion cofactors. Remember, we in the humans, more than 1/3 of all enzymes are such which are requiring metal ions as cofactors. Metalloenzyme. 1/3 enzymes requires metal ions for their catalytic activity. These are metalloenzymes including, and they, in this case, the metal ions are tightly bound to the enzyme for the catalytic activity. Metal ion cofactors are those cofactor, actually we are referring to the metalloenzyme when the metal ion is having the cofactor function for the enzyme activity. Mostly the transition metal ions such as iron plus 2, iron plus 3, manganese plus 2, copper plus 2, they are used as cofactors for the enzyme. These actually explain the need of trace elements in the diet. Actually, from here we come to know that why some elements, trace elements must be there in the diet. For example, iron, copper, manganese, they are actually nutrients and they must be present in the diet. And why? Because look, for here we are explaining that these are actually the cofactors for the enzymes. So these must be in the diet. This also explains the toxicity of heavy metals. How? From here we actually come to know that why some heavy metals may be toxic. For example, cadmium and mercury are the toxic element. Why? Because they can replace zinc. Zinc, cadmium, and mercury belong to the same group in the periodic table, so they can easily replace. So if there is cadmium, mercury in the diet, they can replace zinc. And in the same way, if they replace zinc, the active site of enzymes such as important enzyme of RNA polymerase, active site will be blocked by the mercury or cadmium, and thus we can say that the cadmium, mercury, arsenic, and so on are toxic metal because they are not present in the diet. They are not actually the cofactor, but they can replace the essential elements because of the same belonging to same group. So that's why enzymatic system may be blocked. Metal and cofactor example is we have enzyme carbonic anhydrase. This is a very important enzyme because it is catalyzing the reaction of water with carbon dioxide and it is giving us H2CO3. This is a buffer and a very important reaction because it is maintaining the pH constant of the blood. Carbonic anhydrase enzyme and catalyzing the reaction of water with carbon dioxide, or this enzyme requires the cofactor of metal ion, which is zinc plus 2. Metal ions participate in the catalysis in three ways. There are three possible mechanisms by which these metal ions help the enzyme to carry out the catalyze the reaction. One, they by binding to substrate to orient them properly. First, they come into contact with the substrate, and the substrate is oriented in such a direction so as they are properly arranged for the active site. So the active site is get attached easily, and thus the catalysis of the reaction is taking place. Second, by mediating oxidation reduction reaction. Metal ions are actually ions, though they are accepting electron or they are removing, they are releasing electrons. So that's why they are helpful by oxidation reduction reaction. Whenever there is oxidation, there will be reduction. So they are helping through redox reaction. Another one is the metal ions are helpful. The enzyme to catalyze the reaction. How? They are by electrostatically stabilizing the negative charge. Whenever there is a negative charge in a molecule end, these metal ions gets come together, gets attached, and in this way, the negative charge of that end is stabilized. So metal ions are helping the enzymes to catalyze the reaction. How? Maybe coming in contact to the substrate and to orient them properly, and in this way help the enzyme to get attached to the substrate and catalyze the reaction. They may be carrying out oxidation reduction, accepting electron, or they are providing electron to cause reduction and oxidation reduction reaction. Through oxidation reduction reaction, they are helping the enzyme to catalyze the reaction, or maybe stabilizing the negative charges on molecules. They get together around that region, and this way the negative charge is stabilized. The next type of the cofactor are the coenzyme. They are the organic molecules which are termed as co-substrate because they are transiently associated. The enzyme we are having is succinate dehydrogenase. This enzyme is catalyzing a very important reaction of the Krebs cycle when succinate is converted to fumarate, And the coenzyme required for this enzyme is flavin adenine dinucleotide. FAD in this reaction gets reduced. Hydrogen from the succinate is accepted by FAD, and from succinate fumarate is formed, and this fumarate is the oxidized form, and the hydrogen is accepted by FAD. So this enzyme carry out this important reaction, and FAD as a coenzyme is required. Let's to study the mechanism. This is this flavin structure. This is adenine ribonucleotide, and we have diphosphate link in another FAD. So it means flavin dinucleotide. Flavin dinucleotide. This is flavin. If you look to this highlighted region of the flavin, look here. Here nitrogen double bond, carbon single bond, double bond nitrogen. These are the regions which are actually responsible for the acceptance of two hydrogen. And when it is saturated with hydrogen, look, hydrogen one attached here, one attached here. So flavin has a specific sites to accept hydrogen. This coenzyme is actually helping the enzyme by accepting the hydrogen. Another coenzyme is the lactate dehydrogenase enzyme, and this is also using organic coenzyme cofactor as a substrate. These are also co-substrate because they are transiently available to the enzyme. Lactate dehydrogenase is important enzyme because it is catalyzing the reaction of pyruvate reduction to lactate. Hydrogen from nicotinamide adenine dinucleotide NADH are accepted, and NAD gets oxidized, and from pyruvate we get lactate. This is a very important reaction, especially in the oxygen deficiency, when there is no passing of pyruvate to the tricarboxylic acid cycle. So the only possibility for pyruvate then is to change to lactate, and in this way the NADH are oxidized, so this can be used in glycolysis. This important reaction is catalyzed by lactate dehydrogenase, and again here is the reduction reaction. It requires coenzyme NAD. How? This is the structure of the NAD, nicotinamide adenine dinucleotide. Look this, these are the regions actually which are accepting hydrogen. Look, this is highlighted, and when it is accepted on hydrogen, it becomes like this. So this NAD is a coenzyme, organic coenzyme. This is helping the enzyme, the lactate dehydrogenases, by providing hydrogen. So the enzyme is helped to catalyze the reaction, and this is another type. Third type of the cofactors are the prosthetic group cofactors, and the prosthetic group we already talked about are those ones when the cofactors are tightly bound to the apoenzyme part. Today we are having example of cytochromes, and cytochromes are actually electron carriers, and they contain heme. Cytochromes we know are so many taking part in the electron transport chain. Heme, what is heme? Hemes are actually prosthetic groups of the cytochrome. These cytochrome enzymes require hemes. What is heme? Hemes are having something like this. Hemes consist of porphyrin rings with iron center. Actually iron in the middle, and there is porphyrin ring. This is iron, and this is one porphyrin ring, second one, third. 4 porphyrin rings are linked to the iron. This is called heme. Heme is actually the coenzyme for the cytochromes, and these hemes are actually having organic groups linked to the iron. This is tightly bound. Iron is tightly bound, sometimes covalently bound. So it is not just transiently available. And this iron may be in +2 state, maybe in +3 state, and this explains how it may be helping the enzymes to catalyze the reaction. Heme iron can undergo one electron transition between ferric and ferrous. Iron is changing from ferric state to ferrous state, and thus this is done by accepting electron, and vice versa is also possible from ferrous state to ferric state, and this will be losing one electron. So thus it can help. Look this, iron plus 3 when accept electron it becomes iron plus 2, and vice versa. So it means that iron can help the enzyme by providing electron or by removing electron, and in this way this help the enzyme to carry out the redox reaction. So cytochromes are carrying such reaction in the electron transport chain, and they are having heme which is helping this process for them. They are the prosthetic cofactors. Once again, Western Todd book, GL Jain, and Voigt and Voigt are recommended, especially the Voigt and Voigt. Please highlight each and every point you can see in these books, elaborate and add more good things to your notes, and try to simplify things. In this way, we can. From today, I will say goodbye. Thank you. Allah Hafiz.